Hi, my name is Zach, and I'm a tutor here at Chegg.com. Today I'm going to be discussing with you the topic of protein metabolism. It's a fun topic, a topic that is very close to my heart. I'm going into the medical field, and I hope that you guys can really enjoy this video and learn something. Protein metabolism is a pretty broad topic. There's a lot to it, a lot of mechanisms, and in this video I don't really have the time to go into a lot of the mechanisms. To do that, we'd have to really make individual videos for each subset of these different pathways. So for this video, we're going to have a good, nice, broad background that will get you on a path to really start to understand protein metabolism. Okay, so let's get started. So protein metabolism kind of can be encompassed in this chart. It kind of has like pretty much everything. There are some details left out. Like I said, I can't talk in 10 minutes about all the mechanisms. But anyway, so we're going to start here with proteins from diet. So why do we have to get proteins from diet? So if you think back to the nitrogen cycle or urea cycle, the human body can't make nitrogen, okay? So we can only get it from our diet. And the big, what makes proteins are amino acids. And amino acids, to make those, you have to have amines. And amines basically are nitrogen. So all of our nitrogen has to come from our diet, from steaks or nuts, peanut butter, whatever your diet is. You have to eat proteins to get amino acids. So when you eat these proteins, they get broken down in your stomach and then absorbed in your intestines, and you get free amino acids and absorbed into your bloodstream. Now, depending on what our body's needs are at that time, we can, either go through an anabolic pathway and build body proteins, or, if we need glucose, we can run gluconeogenesis, use that for energy or oxidation. Or if we have so much protein in our body, we just overload it, we need to get rid of it, we can deaminate it and excrete it as urine. Okay? So let's talk about when we'd want to do this anabolic pathway. So let's say that I'm a weightlifter. I'm trying to build up muscle mass in my arms, and so I'm eating lots of protein. Well, since I'm weightlifting, I'm breaking down proteins when I'm bodybuilding, and I need to build, more muscle. Well, muscle is made of protein, so I'm going to take these free amino acids and make those myosins and actins that I need to build muscle and make these body proteins. Okay, just one reason I would use it. Another reason would be maybe, let's say, for your whole life you've lived at sea level in Florida. Well, all of a sudden you decide that you want to move to, Denver, Colorado. You really got into skiing. But when you move up to Denver, Colorado, your body has to adjust for the decrease in oxygen at high elevations. And so what it does is it creates more red blood cells. And more red blood cells means more hemoglobin. And more hemoglobin means you got to make it. Okay? And so what your body's going to do in that case is take these free amino acids, make hemoglobin so you can carry more oxygen to your tissues, and that way you can keep living at this higher altitude without, getting altitude sickness and all those negative things that can come with it. Okay? So now let's say you're in Colorado now, you're out skiing, you get off the trail and you get lost, okay? And you're out there for 5-10 days before somebody finds you. Well, in that time you're out there, you don't really have any food to eat. So what your body's going to do is start breaking down proteins, like your muscle proteins, and turning them back into these amino acids by catabolism, okay? And so once we have these free amino acids, we can then run, these three processes here: deamination, oxidation, and gluconeogenesis. So deamination is going to be central to both oxidation and gluconeogenesis. So let's talk about that first. So in the process of deamination, we're taking, our amino acids, our alanines, our lysines, aspartates, any of the amino acids, they all have the nitrogen. They're not on the side chain, but on the base structure of it. And we're going to take that off by having nucleophilic attack of alpha-ketoglutarate. Okay, so this alpha-ketoglutarate nucleophilic attacks the amino acid, pulls off that amine, leaving a keto acid, which we'll use for oxidation process and gluconeogenesis, and results in glutamate. Okay, and glutamate's one of these big things we always use in deamination, because that side chain is really easy to pull off. Okay, and once we take a water molecule, we can actually take that amine off of the glutamate, regenerating alpha-ketoglutarate and giving us a free ammonia, right? Well, this ammonia is toxic to our body. So then we have to transform it into this molecule called urea, which is basically 2 ammonias, a carbon, and an oxygen. And then we excrete that out of our bodies in urine because it is toxic to us, okay? But we need to run these deamination reactions really to, one, get rid of old proteins that we no longer need. And if we want to run oxidation or gluconeogenesis, we have to do it too. And I'll explain why now. So in gluconeogenesis, we can only do this to the glucogenic amino acids. So that's pretty much all of them except for, I believe, lysine and leucine, I believe. And so we can turn all of the other amino acids back into glucose. Now, why would we want to turn these into glucose? Why not straight into ATP if we're starving, right? Well, you got to think about our brain. Our brain, because of the blood-brain barrier, the only thing that can cross it is glucose. Amino acids can't. So we first have to change these amino acids into glucose so that it can cross the blood-brain barrier, and then we can run glycolysis and Krebs cycle to then break that glucose into, break it down and make ATP to fuel our brain, okay? So that's kind of why we run this gluconeogenesis process and why we change the amino acids to glucose. Anyway, so why do we need to deaminate these amino acids to do this? We deaminate the amino acids because in the gluconeogenic cycle and the Krebs cycle, you don't see amino acids. But if I'm to take an amine off of it with a deamination reaction, or move an amine from one place to another via a transamination reaction, I can make the amino acids then into substrates that are common in the gluconeogenic and Krebs cycles. Okay, so that's why I need this deamination for gluconeogenesis. The same thing happens when I take amino acids and oxidize them. These are typically the ketogenic amino acids, or your lysine and leucine. And in these reactions, what I'm going to do is first turn these amino acids into lipids via deamination, transamination reactions, and then I'm going to break down those lipids and make energy. Same kind of idea I'm having with the glucose here. Okay, so that pretty much summarizes protein metabolism into a nice little compact overview. Like I said earlier, there's a lot of mechanisms involved with these reactions, and for the scope of this video, I really don't have time to go into those. So if you would like more details on that, please request that video from Chegg, or contact the tutors to and let us go over that with you. Anyway, I'm Zach, tutor at Chegg, and I hope that you have a wonderful rest of your day.